now we're going to look at a very important practice in labs all over, science labs, chemistry, biology, whatever, dilution of solutions, all right? So this is something that is necessary very often because very often what you'll find, okay, this thing is not, okay. So very often what you'll find is that you might have an experiment which requires a solution of a particular concentration. However, at hand, what you have in the lab would be a solution, but the concentration would be higher than what is required, right? So what you have to do is basically dilute the solution to the extent that you need. And that means that you need to know exactly how much solvent that you need to add. Because after all, as stated here, dilution is a process of making a more dilute solution by adding solvent to the more concentrated solution. The key thing you need to remember about any dilution process is that when you add the solvent, you're not changing the amount of solute present. What you're doing is increasing the volume. And by so doing, you'll be changing the concentration by decreasing it basically. So as stated here, it is very common practice that in the labs, if an experiment requires a more dilute solution, then what we would do is dilute a more concentrated solution. So very often what we do is that we prepare a concentrated solution and then dilute it as needed based on whatever experiments that we're trying to carry out, all right? So just to visualize what's happening when you're diluting a solution, here we have this diagram right here. So initially we have a solution which contains 0.0050 moles of iodine in 0.1 liter of solution. So therefore this would be the resulting molarity, 0.050 m for the iodine solution. And then what this person did was add water to it until the final volume increases to 0.5 liters. Please note that the amount of solute, the number of moles of solute, remains the same. But what happens as a result of increasing the volume fivefold is that you have now diluted the solution fivefold. So you'll see that the concentration of the solution after dilution is 1/5 of what it was before, because you have basically increased the volume of the solution fivefold, all right? So if you could actually see the particles in the solution, you'll see that, for example, over here, you'll see that within a certain volume, you'll have a certain number of iodine molecules, but as you dilute the solution, you see that within the same volume, the number of iodine molecules decreases, all right? So that's basically what happens when you dilute the solution. Basically, the number of solute particles are more spread out, and therefore, the amount of solute particles per volume would decrease as a result, all right? So that's basically what happens. So when it comes to calculations, it's a very easy equation that is used. It's based on what we said before. The amount of solute does not change. So that means that before and after dilution, the product of the molarity of the solution and the volume of the solution will remain constant, because the molarity times the volume gives you the moles, and the moles does not change upon dilution. So therefore, the result of that is that there's going to be an inverse relationship between the molarity and the volume, right? So the molarity, or the concentration, times the volume of the more concentrated solution will be equal to the concentration times the volume of the more dilute solution. That's basically the relationship between the concentration and volume of the more concentrated solution and the same values for the more dilute solutions. Now, me personally, I prefer to use this equation here. It means the same thing, so there's no real difference. So the equation I prefer to use is C1 V1 is equal to C2 V2, right? And let me just say here that for this equation, it doesn't matter what the volume units are, right? So the volume units for V1 and V2 could be liters, it could be milliliters, it could be gallons, it doesn't matter, as long as they're the same. And the same is true for the concentration units. So C1 and C2 can be expressed in different concentration units. Of course, the one that we are focused on in this chapter would be molarity. So if this has the molarity units, then C2 would have the molarity units as well, all right? So just to give an example as to how this equation works, Here's an example right here. Question says, how many mils of a 2.00 m copper 2 sulfate stock solution are needed to prepare 0.250 liters of a 0.400 m copper 2 sulfate solution? So the equation we're going to use here is C1 V1 is equal to C2 V2. And when you're given a question like this, of course, you have to identify the different terms. So based on the information provided here, the question is asking how many milliliters of the 2.00 m solution. So the question here is asking for V1, right? C1 would be the 2.00 m, V2 would be the 0.250 liters, and C2 would be the 0.400 m. So since the question is asking for V1, I would rearrange this equation to solve for V1. V1 will work out to be equal to C2 V2 over C1. And all I have to do from here is plug the numbers in. So C2 is 0.400 m. V2 is 0.250 liters. And C1 is equal to 2.00 m. So these will cancel out. And therefore, the answer, let me calculate this, 0.400 times 0.250 divided by 2.00. And according to my calculations, this works out to be equal to 0.050, how many sig figs? 3 sig figs, so another 0 liters, all right? So this would be the volume of the more concentrated solution that is the 2.0 m copper 2 sulfate that you need to measure in order to make 0.250 liters of a 0.400 m copper 2 sulfate solution. And the pictures that you see here below is basically demonstrating how this is actually done in practice. As I said before, this is done in labs all over where we want to make a solution that is more dilute of a specific concentration. So basically, in order to do this accurately, we use this piece of glassware. This is known as a volumetric pipette, all right? This is made very accurately, and they come in different sizes. The one that you see here on the screen is a 50 ml pipette. It has a mark up here, which you cannot see, but basically, if you pull the liquid into this pipette up to that mark, where the meniscus gets to that mark, then you would have measured 50 mils, right, of that solution. So this solution that is in the bottle here is the original 2.00 m stock solution of copper 2 sulfate. This person withdrew 50 mils of the copper 2 sulfate solution and then dispensed it into a volumetric flask, which presumably would be the 250 liter, or I should say 250 mil volumetric flask, which is 0.250 liters, right? So basically, the copper 2 sulfate that was measured, the 50 mils of the copper 2 sulfate that was measured, would be dispensed into the flask, and then water would be added to this flask until the water level gets up to the meniscus, which represents 250 milliliters of the solution. And that's it. And that's how you basically make the solution. The only other thing that you need to do is to shake it up a bit and make sure that the solution is homogeneous as a result. Okay, so that's basically how we do a calculation of this type. Okay, so the next topic that we'll be talking about in the next video is where we're going to combine what we learned in this chapter with respect to molarity and what we learned earlier in this chapter with respect to stoichiometry, and we're going to see how there are problems that you could do which combine the concepts involved under those two topics. All right, so that's it for this video. So until next time.